Thank you for everybody for inviting me for this absolutely fabulous meeting. So I'm going to be talking about liver cancer. It's not a Mutographs cancer, but it's the second leading cause of cancer death worldwide. And it turns out that mutational signatures have revealed a previously unexpected but widespread carcinogenic exposure, and that exposure, as you might guess, is the exposure to aristolochic acid in liver cancer. So I'll talk about AA compounds in herbal medicine. I'll talk about the AA mutational signature and urothelial cancer. And then I'll go to the main event, which is AA and liver cancer. And then I'll try to summarize what I've talked about and talk about where we might go from here. So there are many compounds related to aristolochic acid. Here are a few of them. Right now, a few compounds have been studied intensively, basically AA1, which is the compound on the upper left, AA1. It's been the most intensively studied. David Phillips talked a little bit about AA2 earlier this morning, and so his data suggests it has a different signature, but it's a signature we haven't seen in cancers. But for the purposes of my talk, we're going to lump them all together as AA. So many herbal medicine traditions use plants containing AA, and this occurs, AA occurs naturally in these plants, so it's not an adulterant. People don't add it for medicinal effect. So these are plants used in traditional Chinese medicine. Most of these plants are in the genus Aristolochia. This plant is in the genus Azarum. They're both in the same family. And actually, I brought some. So I'll tell you about what I brought. Yeah, pass it around, please. Yeah. So what I'm passing around is number one is this fibery one is a root. It's shishin. It can be from several different species. It's very hard to tell what species it is. And I'll talk about the other one I'm passing around is guangfangji, and I'll talk about that in a little bit of detail later. And then the third thing I passed around is a sachet of dry powdered shishin root, okay, which is shishin is always a root. So these are genus Aristolochia and Azarum. But it's not just Chinese herbal medicine. So they're used in Romania. So I can't really read from here, but I think it's good for cancer, ulcers. I think this is hair loss and hemorrhoids. And it's used in Brazil. This is Aristolochia. I can't read it. Symbifera. I can't read it. Sorry. This is another species in genus Aristolochia. It's used in India. This is Aristolochia indica, and the part they use in Ayurvedic medicine in this case would be the seed pod, which is this green oval here. Okay. So it's not just Chinese medicine. It's used in herbal medicine traditions in Latin America, Africa, I don't have an example, and to some extent in Europe. It's used for many indications in traditional medicine. So it's like Romania, it's good for a lot of different things apparently. So aristolochic acids metabolize to aristolactams and then acylnitrinium ions that form DNA adducts on adenine. They form covalent bonds on adenine, and this leads to a lot of adenine to thymine mutations. They were well known as Nefer Toxins back into the 1800s. So here they were given to rabbits and caused kidney failure. Here is horses being poisoned by eating the European species. It became known as a human public health problem in the 1900s. About 100 young women with end-stage kidney disease going to the same weight loss clinic in Belgium. And what had happened is they were using some Chinese herbs. They changed powdered hanfangji with powdered guangfangji. And this one is a totally different genus and doesn't contain any AA in it. and this one contains AA. And many of those patients with kidney failure then went on to develop upper tract urothelial cancers, a relatively rare cancer in most situations. So this is just the innocuous one, the Hanfangji, no AA, and this is the one that contains the AA, and it's the one that I'm passing around. So this is the Guangfangji. And in fact, the herbal medicine tradition doesn't make a strong distinction between the two medicines. And so in fact here it just, anyway, that's the main point. The herbal medicine tradition doesn't make a strong distinction between the two roots. So now to go back to signatures, I thought okay, I'm going to skip over all this actually, because I thought I'd actually canceled it. Okay, so here's the mutational signature of an AA-exposed upper tract urothelial carcinoma, right? So we've seen this many times this morning. So each bar represents, each bar represents the number of CAG to CT mutations across the entire genome. So the higher the bar, the more the mutations. And here's where upper tract urothelial carcinomas occur. So they're right here where the tube that drains the kidney joins the kidney, and that's where these tumors are forming. Again, that peak is CAG to CTG peak. Then this peak is a CAG to, an AG to ATG peak. So there's not as many of those mutations. And then there's hardly any mutations from C to A, C to G, C to T, and so forth. And here's what an AA-exposed cell line looks like. So it looks very, very similar. And in fact, this is actually lowly mutated. And so the differences here are mostly due to just sampling variance and this relatively low mutated cell line. So a few years ago, in 2013, we knew about upper tract urothelial carcinoma. We knew that that had the AA signature, and we knew that AA-exposed cell lines had the AA signatures. But then by 2 years ago, anecdotally, we had seen the AA signature in liver cancer, in bile duct cancer, in kidney cancer, and in bladder cancer, we and other groups. And so we decided to look at AA in liver tumors in more depth. Oops, I'm using the wrong button. And it turned out that it was known that Taiwan is a known hotspot for AA exposure, but AA had not been intensively or specifically studied in Taiwan. So we looked at 98 liver cancers in Taiwan, and we found to our shock that almost 80% of those liver cancers had very strong evidence of AA exposure. So in this graph, the height of the bar represents the number of mutations in an exome, and so the dark red bar here is the AA mutations, the mutations ascribed to AA. So that's the spectrum of one of the tumors. That's the spectrum of another tumor that contains AA and something else that we don't know what it is actually. Here's the spectrum of a tumor that doesn't have any AA in it. And it turned out that liver cancers have been pretty widely sequenced. So we were able to ask the question of how extensive is AA exposure in about 1,500 other liver cancers around the world. And this is the short answer. So basically, Taiwan remains dramatically the hotspot, but mainland China had prevalent exposure. Korea had quite prevalent exposure. Southeast Asia had quite prevalent exposure. Japan, not very much. One patient out of 5 from Brazil had AA exposure. He was not, I believe it was a he, was not Asian. North America, not very many, except for the Mayo Clinic. Europe, not very many. We think that these patients were all listed as Asian, and they had no country information, so we believe that we speculate that they came to the Mayo Clinic for treatment from Asia. Speculation, right? Just so you can see the primary data a little bit, these are the spectra. So again, in most cases, what you're seeing here looks a lot like what we saw in the upper tract urothelial carcinomas and a lot like what we saw in the cell lines. The number of AA mutations was extremely variable. Taiwan was close to the champion. Mainland China, even with almost 50% exposure, had much lower levels of mutations associated with AA, and then some of the other countries were in between. I also should mention that since this paper was published, about 50 out of 100 combined HCC and bile duct cancers had AA exposure from mainland China, and in another area where there's a lot of aflatoxin exposure to another known liver carcinogen, and then a competing hazard, we still had 25% of these tumors had AA exposure. So obviously, this leads to the hypothesis that AA exposure elevates risk of liver cancer, and then I would speculate that this could be synergistically with hepatitis B infection. So the evidence, AA is a known mutagen and urinary tract carcinogen. We found multiple known cancer driver genes with AA mutations in these data. And the proportion of AA-exposed liver cancers in Taiwan at 78% is a lot more than the 33% estimated the proportion of the population exposed to AA herbs in an epidemiological study. Since then, there's been an epidemiological study in Taiwan where there's national health insurance, so they found a linear relationship between AA dose and risk of liver cancer in hepatitis B infected and hepatitis C infected patients who visited traditional Chinese medicine clinics. So you can do this study in Taiwan because the national health insurance covers traditional Chinese medicine. And then just in December, a laboratory, Zhuang Han in Shanghai, showed that you can give mice liver cancer by exposing them to AA at 14 days. So the slide's a little funny, but basically, for low levels of exposure, you get only a small number of tumors very late. For very high levels of exposure, so that would be this column. On this column, you get a lot of tumors. The reason there's no star on this bar is that the number of mice that survived that long are very few. But so now there's experimental evidence that AA can cause liver cancer in mice. So to try to summarize a little bit, the main results are that liver mutation data indicate widespread exposure to AA in East Asia. AA is a known urinary tract carcinogen and kidney toxin. The prevalence of AA-mutated liver cancers varies by geographic location, but is high in many parts of East Asia. And AA herbs are also used in Africa and Latin America, but AA exposure in cancer there has not been studied. The number of AA mutations in affected tumors varies by geographic location, but multiple lines of evidence, including the epidemiological evidence from Taiwan and the experimental evidence in mice from Professor Han's laboratory in Shanghai, provides strong evidence that AA causes, i.e. strongly increases the risk of liver cancer. So this could lead to further research that could lead to prevention. So we need to know what's the dose-risk relationship between AA exposure and liver cancer, and in conjunction with or without hepatitis. And we're also interested in developing non-invasive tests for AA exposure. We also want to know which of those chemicals that I showed in one of the first slides and which doses have the main impact on kidney failure, urinary tract cancer, and liver and bile duct cancers. And we also really don't know how people are being exposed. We don't really know which herbs people are using and how they're being prepared. And in fact, some of the compounds, the aristolactams, are quite prevalent and haven't been studied at all. We don't know if they're bioavailable. And actually, they occur in other species besides the ones that I mentioned. And then what's the extent of exposure in Africa and Latin America? And again, what are the toxicity profiles of the various AA-related compounds? So I just want to point out that even within one part of one species, the compounds that are present is extremely variable. So this is Guanmutong, which is the stem of Aristolochia manchuriensis, and the columns here, the 1, 2, 3, 4, 5 columns, represent different specimens of the stem of one species, and each row is a different AA-related compound. So AA1 here is at the top, but then there are other AA-related compounds, and you can see that the profile of different specimens is extremely variable. And there's no one actually really working. This is from Johanna Michel, who was a graduate student, I think, at University College London, and has gone on to another area now. And as far as I know, no one is actually studying this question at this point. Here's another one part of one species. So this is Tianshentang. So that's the thin above-ground stem of Aristolochia debilis. And again, you can see that the profile of compounds present in the two specimens was very different. So we can talk about prevention through secondary prevention, through screening. We can develop non-invasive tests, we hope. So identify patients for screening, for cancer screening, or at risk of kidney failure. What's the risk of kidney failure after an AA-associated cancer? We could think about therapies. High mutation loads might correspond to better response to immune checkpoint inhibition. More speculatively, elevated missplicing in these tumors is common because CAG, all our introns basically end in CAG. And so there's a lot of missplicing in these tumors. So that leads to elevated nonsense-mediated decay. So that might represent a vulnerability in these tumor cells. I'd like to also bring up some other issues that I just sort of was thinking about at the end, right? So how do we convince funding agencies that further research is important, and what could be the role of patient advocacy in this? In a lot of these countries, I think there hasn't been a large tradition of patient advocacy, and so maybe it's possible. So I'm actually quite concerned about education, you know, as the way to improve prevention here. But there are very important issues around cultural pride. So this is traditional Chinese medicine, and Chinese suffered 100 years of humiliation at the hands of the West. And there's also the interests of the herbal medicine industry, which is a very big industry. So with those issues unresolved, I want to just acknowledge my colleagues at Changgung Memorial Hospital in Taiwan, and Alvin and Song Ling and Arnaut and Tay Bin Tian, Patrick Tan, Alex Chang in Singapore, and Jacob Pang at Changgung Memorial Hospital. And with that, I'd be pleased to take some questions.